Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be going through the AQA A-Level Biology Paper 1 from June 2018. This is the paper that I actually did when I did my A-Levels, so it's going to be a lovely blast to the past. Figure 1 shows all the chromosomes present in a human cell during mitosis, and the scientists stained and photographed the chromosomes. In Figure 2, they've arranged their images of these in homologous pairs. So homologous pairs, actually, this just while we're here as well, is the maternal and paternal copy. So if you have homologous pairs, you're a diploid organism. There's 2N. A lot of people make the mistake of saying homologous pairs separate. In mitosis, the homologous pairs do not, because we have 2 copies of every chromosome. And if you're saying the homologous pairs are separating, you're saying that the maternal and the paternal ones have split. So then you have daughter cells with 1 copy of each, which is not correct because that's haploid. So just as a quick thing here, the homologous pairs only separate in meiosis because you're making haploid cells. In mitosis, the homologous pairs do not separate. These are not sister chromatids either. So sister chromatids do not exist as maternal and paternal like this. That's also incorrect. The sister chromatids are copies of each one. So maternal and maternal, and then for the same one, it would be paternal and paternal. So based on that, if these were sister chromatids, it would look like this. So there's 2 of this and 2 of this. So 2 maternal, 2 maternal, 2 paternal, 2 paternal. That's something I see people just fumble horrendously. The definitions are there. They say that you make a diploid organism. If you are diploid, you've got maternal and a paternal chromosome for every single one. So by definition, if you're doing mitosis, you're going to have to find a way of making sure those cells have 2 copies to be genetically identical. It says, give 2 pieces of evidence for Figure 1 that this cell's undergoing mitosis and explain your answers. So number 1 is that when mitosis is happening in your cells, you've got mitosis, and then you've got your interphase, which is every other step. Interphase. You've got G1, gap phase 1, synthesis, gap phase 2. Now, gap phase 1, cells getting bigger, lots of organelles. Gap phase 2, cells get bigger, lots of organelles. Synthesis is where DNA replication happens. Now in mitosis, the first step is prophase, where the chromosomes condense and the nucleus, the nuclear envelope breaks down. So already, number 1 is that they're condensed. You can actually see them. Individual chromosomes are not usually visible. They're kind of like long, stringy bits and bobs like that. So in this case, number 1 already we can say is that the individual chromosomes are visible, because you can't just say that because that doesn't explain why it's mitosis and doesn't explain the answer. It's because they have condensed. You can't just say that they're visible. That's just saying, okay, that you're just like, if I said, like, I don't know, explain why buying a really rubbish car is a bad idea, and then you say you lose money. Well, you lose money buying a car anyway. You would then explain, being like, oh, well, you'd have to spend more money repairing it and this and that. So here it's saying, explain why you can see it's undergoing mitosis. The thing that you're picking up, the evidence, is that you can see the chromosomes. Why? Well, they must have condensed, which happens in mitosis. Now, another piece of evidence is if you look closely at these pairs, this picture's a bit unfair because these are already sister chromatids as well. Normally, if you had your normal maternal and paternal, it looks like this. I'm just adding this now because I've realized from before I've misread that part. Right, maternal paternal. When you're doing mitosis, you're making maternal with a sister chromatid and a centromere in the middle, and then you've got a paternal like this, like so. Here, you see there's 2 pairs of every chromosome, but if you look closely, they are already sister chromatids bound by a centromere. It's kind of hard to tell. So that's another thing as well, is that each chromosome is made of 2 chromatids. So again, that's just your observation. You need to say why. Because DNA has replicated. So it's really important to understand that as well. So in mitosis, again, your cell's chilling like this, maternal, paternal. When you've replicated it, you get maternal, centromere here. Paternal, centromere here. Then the cell divides and you get maternal, paternal, maternal, paternal. Otherwise, if the homologous pairs, which are these, separated, then you would end up with haploids. So it's 2N. You've got double 2N here. So it's not 4N, because 4N would suggest 4 parental chromosomes. This is like paternal, maternal, and then something else. It's 2 lots of the 2N because it's uncopied. And then we're back down to 2N again. In meiosis, the homologous pairs basically mean pairs from the same chromosome from each parent. So if so what happens in meiosis is the same thing. So maternal, paternal, normal cells chilling, right? 2N. Then what happens is you get DNA replication. So again, maternal, paternal. That's still diploid. We're at 2 times 2N, but meiosis makes haploid. So then what happens is the homologous pairs separate. So meiosis 1 separates maternal-maternal and then paternal-paternal. So you can see now we have 2 times haploid, because there's only paternals here. They will have been crossing over and to sort of exchange alleles and independent segregation as well. And then in meiosis 2, these chromatids separate. So you've got a maternal, and then you've got a maternal, and you've got a paternal, and then a paternal. But just remember that crossing over happens here. So these are all haploid. Sorry, these are all haploid. Whoops, N. N is just the number of homologous chromosomes you have. So if I've got 4N, then that means I could have like maternal, paternal, something else, and something else. So it's really important to make sure you understand that the homologous pairs are mother, father, or maternal, paternal chromosomes. Not anything to do with mitosis. The homologous pairs are still always there. Another piece of evidence you could say based on this figure is that the homologous pairs, in this case, are not arranged in homologous pairs. Like, they're not together, they're not all lined up. You can see the homologous pairs here because they're nicely spread out, but here it's not. If it was meiosis, then they would be, because then that means we could split them, like, you know, into 2 different cells and then split the maternal and paternals, making haploids. So it's really important to get that clear. So then it says, tick one box that gives you the stage of mitosis shown in figure 1. So if we look at figure 1, what's going on here? So they're just condensed, that's all. So it's going to be the start, prophase. They're not lined up in the middle. Metaphase, middle phase. Meta means middle. So they're not lined up at the equator. They're not all lined up in the middle part. So that's out. Anaphase, they look V-shaped because they're being pulled across. Telophase as well. They'd be 2 separate like groups of all of them, like, you know. So there'd be single chromatids in separates. So like, you know, like on separate sides. So each one of these would now, you'd have like, each cell would have these, but like as a singular chromatid instead. When preparing the cells for observation, the scientists put them in a solution that are slightly higher or less negative water potential than the cytoplasm. This did not cause the cells to burst, but move the chromosomes further apart in order to reduce the overlapping of the chromosomes when observed with an optical microscope. Suggest how this did that. So it's suggest. So they've just told you that this person, what they've done is they've used a slightly higher water potential as the solution. So cells in higher water potential solution. So the only thing that can explain what might be going on is that water moves into cells via osmosis. And they've told you that this thing moves the chromosomes apart. So you need to think of any possible reason as to why water moving into a cell would move the chromosomes apart. So what it must be is, the only explanation is that, well, the insides of the cells are getting bigger and the chromosomes are spreading out. So the cytoplasm gets bigger in size, or you could say the inside, you know, spreads out, the volume of the cell increases. And that makes sense, because if I've got like cell here chilling, these are the chromosomes all nice together, and I add water and it's like nice and spread out. This is exaggerated. They're nice and spread out. That's the only explanation you can say. So just make sure you remember to work with the question what it's saying. The dark stain used on the chromosomes binds more to some areas of the chromosomes than others, giving them a striped appearance. So just one way the structure of the chromosome could differ along its length to result in the stain binding in more than one area. These are both chromosome 1. So if I just take that and move it over here, like if I zoom in on it here. Chromosome 1, maternal and paternal, right? Now, there's darker here, and there's some darkers there and darkers there. So you've got these like darky looks here, and then a bit of dark and a bit of dark there. These are the same chromosomes, so they should have the same genes, right? If they're the same genes, for example, then, you know, surely they should all be the same. However, they're not. It's binding to some more than others, and it's giving that appearance. So if you look between these a bit more closely, any gene in the universe on a chromosome is like a section of it that codes for a specific protein. So if I looked at the maternal and the paternal ones, 1 and 1 here, the genes would both be at that locus here. A locus is where a gene is found. So then, if they're both making the same gene, so for example, if I say, like, I don't know, hair color protein, I'm making this up because I don't actually remember how hair is coded for. Why on earth do people have parents with like blonde and black hair and the kid is black haired? Well, these genes are both coding for the same protein, but they're different in how they're expressed. The expression is actually different because you've got black hair versus blonde hair. So they're the same protein, but they're different versions of it, so those are alleles. But they're the same gene, so where's the alleles in this? If you zoom in on the genes, it will have in the base sequences of like, I don't know, A, T, C, C, G, G, whatever, right? And then, I don't know, A, G, C, C, G, G. These are a section of DNA coding for it. This is made up, but those would be a gene, and that's also the same gene. But you can see that their base sequences are different. So what makes an allele is the fact you have the same gene, but there's just different base sequences. What determines what a gene is where it is found on a chromosome, its locus. It doesn't matter, you know, if it's my chromosome 1 or your chromosome 1, as long as we don't have too many mutations going on. And your chromosome 1, the gene here, will also be the same as mine. Your mother's one will be this, your father's one will be this, and so forth. Like, all of it, the gene is literally just where it's found, and it leads to a protein. The alleles are differences in the base sequences. So back to this question then. It's telling you that it's for some reason binding on more areas than others, giving them a striped appearance. So in theory, these should be identical. The only difference between them, like I said, is that they must have, well, they've got differences in base sequences. And that must be the reason that it's binding differently. And that's because of those alleles. So then it says, figure 2, they are arranged in homologous pairs. What is a homologous pair of chromosomes? So it's 2 chromosomes carrying the same genes. A whole load of genes together, and a big thing is a chromosome. Same genes. Homologous, for example, if this is you, and this is me, this is your right arm, that's my right arm. Our right arms are homologous. They're both right arms. Yours is just different to mine, but they're still homologous because they represent the same thing. So homologous chromosomes are chromosomes that represent the same thing. The representing they're doing is carrying the same genes. The alleles are different though. So now it says, give 2 ways in which the arrangement of prokaryotic DNA is different to the human DNA in figure 1. Well, first of all, that prokaryotic DNA is circular instead of linear in eukaryotes. As well as that, there are no histones. Other things you could say as well is that we have one big molecule of DNA in plasmids, prokaryotes, and they could also have plasmids too. A student investigated the effect of surface area on osmosis in cubes of potato. They cut 2 cubes of potato tissue, each with a side of 35 millimeters. They put 1 cube in a concentrated sucrose solution, the other one in 8 equal size small, they cut the other one into 8 equal size smaller cubes and put them in a sucrose solution at the same concentration for the large cube, and then they recorded the masses at different intervals. So the results are shown in this. So whenever you do these questions, just have a quick second to look at what's happening between the 2 things. As one goes up, one goes down, things like that, right? So when you do it one by one. You want to be systematic, because what you might do is you might jump from here to here, and then, oh, here, and then here. Like, don't do that. Do it one by one. So rather than being like, oh, well, there's this, and then there's this, and then like, oh, actually, but this and this, like, start by one thing at a time. So I'm going to look at entirely just line A, then line B, and then the differences between the 2. If you do it like that, you will never miss anything out. So I can see that the total mass is on the y-axis, the time is in minutes. But in A, the total mass is decreasing here quite quickly, but then it starts to slow down in how much it's decreasing over time because it's less steep. In B, I can see it's decreasing quite quickly and also following the same pattern, it's starting to level out. Right, so I've done A, I've done B. Now if I look at A versus B, at the start, B's decreasing in mass faster than A, right? But it seems to start leveling off at the similar points. And then at A, it levels off at like a 57-ish. And then this levels off at 53. So when I said same point, sorry, I meant as in time-wise. But you can see here that it's still pretty steep versus A. So those are things that you should just sort of quickly run through in your head because it'll help you understand what's going on. And that's for the 1 large cube, and that's for the 8 small cubes, which kind of makes sense. That's what you kind of expect because of the surface area stuff. So then it says, describe the method the student could have used to obtain the results in figure 3. Start after all the cubes of potato have been cut, and then consider variables that should have been controlled. So whenever you're describing an experiment, rule number 1 is to figure out where your dependent and independent variables are, and then the rest falls into place. Because then you'll know exactly what to do. So the independent is what you're changing, and dependent is what's being measured. Dependent means it's depending on the independent variable to change. So it doesn't mean just like go back and repeat what it says, because then you're saying, oh well, the independent variable is a surface area, and the dependent variable is osmosis, because that's not what's been reported in the table. What I'm saying is that you've got to look at the independent and dependent variable as in what you're actually measuring and looking at. So the independent variable that they're changing is the surface area of the cube by cutting it, right? Now, the dependent variable is the mass decreasing. So can you see how that's not? Dependent is osmosis. And this, I mean, that's still surface area, but still, like, if you do that, then you know exactly where to start going with your method. Because now, what you're changing is the surface area of the cube, and you're looking at how much the mass decreases, and I should really say, over time. And now the method becomes quite easy, and every step you should think about what needs controlling. So then, they've already done the first part of describing how we get the surface area of the cube sorted out. We need to talk about how we're going to do the next parts. So you should think here, well, I've changed the surface area of the cube. I want to see how that changes over time. Oh, I'm going to have to measure the mass. So you need to measure the cube mass before putting in the solution. And then you should think, okay, well, what could go wrong? What could screw this up? What can I control? Well, you should dry them as well beforehand to remove excess water with light paper towel. Then, once you've dried them, you're going to now put them in, put them in a sucrose solution. And then you need to think, okay, well, what do I need to control? Well, first of all, I mean, we're looking at, again, how the surface areas are changing it. But remember that osmosis can change due to temperature as well. So we need to keep temperature constant. So you could use, like, I don't know, a nice water bath. What else can you think could go wrong? Well, it's looking at surface area of the cube. If I plunk them right at the bottom of here, that surface is not being covered. So suspending them, cubes, to ensure all surfaces contact water as well. Also, sorry, the sucrose solution. And then as well as that, the data shows that it's been done in times every 20 minutes. So then record mass after drying as well, like before with paper towel, every 20 minutes using a stopwatch. And that's it. Like, again, every experiment in the universe, you should look at the dependent independent, but not just like the overall experiment, as in what you're actually looking and doing and measuring yourself. We're not looking at osmosis directly. We're seeing how mass changes to then conclude what could happen with osmosis. The loss in mass shown in figure 3 is due to osmosis. The rate of osmosis between 0 and 40 minutes is faster than in B, which they've actually told you, which we figured out anyway. Is the rate of osmosis in millimeters squared per minute different between A and B during this time? Use appropriate calculations to support your answer. So, this is rate of osmosis per millimeter squared per minute. So the rate's going to be our gradient, right? And that's going to be our your per minute part. But you can see it says per millimeter squared. The per millimeter squared part means that for every 1 millimeter squared of this, you'll get this. So what we need to do is we need to divide our gradient by the surface area of the cubes. So to work out the gradient, it's going to be y2 minus y1 over x2 minus x1. So one problem I see a lot of the time, errors people make, is that they'll say like here and here, and they'll say that's y2 and that's y1 just because it looks nicer calculations wise. You don't get a negative number. That's wrong. This is your y2, that's your y1, because you read from left to right. Now that's important because you might have things where it's asking for a negative gradient. Otherwise you'll get a positive answer, which suggests the gradient's positive. So for A, the gradient is going to equal y2 minus y1, so 57 minus the what? y1, which is 61.8 divided by 40. If I do that, I get, so that's what, 0.12 for that one. And then for B, the gradient is going to be 53 minus 61.8 over 40, which is 53 minus 61.8 divided by 40 is 0.22. Fine. Then we need to work out the surface area of this one large cube for A. So that's going to be, well, A is 35 by 35, and it's a cube, so the depth is going to be 35 as well. The surface area of a cube is you work out the area of one side, and then you times it by 6 because there's 6 sides to it. So the A surface area's cube is going to be 35 times 6, which is 35 times 35 times 6, 7,350 millimeters squared. So this is where it gets confusing. Imagine this is the original cube. If I cut this cube in half, like if that's side on and I slice it this way, and you get this. If I then cut it like this way, then I get this. So you would end up with, so originally you'd have 1, 2 weird cube shapes, right? Now you would have 1, 2, 3, 4 rectangular cuboidal things like that, right? But to make it even inside, the student must have put a cut there, and also a cut here as well, and there. That's 1, 2, 3, 4, 5, 6, 7, 8, like if you picture the other ones at the back. So that's how you get the 8 cubes. Now if that's the 8 cubes, you can see that the length of these is actually 35 over 2. So that means these cubes' surface area is 35 over 2 times 35 over 2 times 6, but remember there's 8 of them, so it's going to be times 8. Now if you do that, B's surface area is 14700 millimeters squared. So therefore, the rate per millimeter squared for each is 0.12 divided by, where has it gone? 7350, and the other one is 0.22 divided by 14700, which should give you, let's see what happens, eh? So 0.12 divided by 7350, it gives you 1.63 times 10 to the minus 5, And then for the other one, 0.22 divided by 1.0014700, you get 1.496, which is roughly like 1.5 times 10 to the minus 5. So yes, there is a difference. So bees are flying insects that feed on nectar made in flowers. There's many different species of bees, So scientists investigate the biodiversity measured in 3 different habitats during a year. They collected bees from 8 sites of the habitats 4 times per year for 3 years, and the scientists' results are shown in the figures that they presented them. So again, just have a quick look at what's going on. So the natural habitat, you can see in what's going on here. So the mean number of bees goes up as you hit like 100 days of the year, and then the mean number seems to go down later on in the year. That's a natural. Then in this graph, if you look at the day of the year and it's the mean species richness, the species richness tends to go up at about day 140-ish, and then it goes down. Fine. If you look at town, so town's kind of going up and down as well. Farmland's also going up and down. However, the species richness in the town is constant. It's not changing. It's the same. Whereas in farmland, it goes up and then it comes back down. Then we compare between each other. So remember, line by line, then with each other. So if you look at natural, there seems to be this huge shoot up in the number of bees and down naturally. In the town, they go up and down, but not as high. They start lower and they finish lower. And in farmland as well, again, they start lower, exceed the point of town, and then go down. And then for the species richness, naturally it goes up and down. At all points, it's higher than the other two. In the town, again, the species richness is completely just the same. And then in farmland, the species richness goes up, overtakes the town, and then levels off at around the town as well. Fine. So then it says, what is meant by species richness? So it's just the number of different species in a community. You can't say population, because the population is all of these numbers of individuals of each species. So for example, if I have woodland and I look at, for example, a very specific type of bird, and the population would be that bird within that ecosystem. So a community is individuals of all these different species within a certain environment. So from this data, the student concluded these things. It said the natural habitat's most favorable for bees, and then the town is least favorable for the bees. Do the data support this conclusion? Explain your answers. So here then, let's have a look. So if you look at natural as best, like if we look at what's for it, so we can see that for the mean number of bees, it peaks, here and then goes back down, but across the board, it's highest. It's higher than every single curve except for the town, right? That's the number of bees, whereas the number of bees, the species richness is always higher than every other one as well. So then, first of all, I mean, you can say that the mean number of bees in natural habitat peaks higher than the other two. I'm too lazy, you write the others, but I can write the other two. But also is higher until day 200. So that suggests that, like, up to this day, they're quite happy being there, or else why did their numbers go up, right? But then as well as that, if we look on the other one, you can see here, like I said before, that's why reading the graph just real quick beforehand helps. The mean species richness is always higher, right? However, the problem is that like after day 200, it drops below both the others. You can see that kind of goes against that. So then town worst for the town, if we look here, we can see that the mean numbers is going up and it's down, but it's above farmland here and kind of hits like a peak that's similar to it. So kind of can't really get anything out of that really much. However, if we look on like the other one here, we can see that the farmland peaks up here and this peaks up here. So remember the interline comparison where it's completely unchanged throughout. So species richness unchanged and natural plus, oh my god, I've written it above there again. Sorry, it's supposed to go down here. But natural plus farmland peak higher. So that's because remember, they're going to change over the years as because we're getting cold and this and that. Bees don't like living in the cold, this kind of thing, right? So we kind of have to look at the picture overall. So there's that. But then we need to look at some againsts. Well, the species richness, it's got a higher species richness than farm, like here, doesn't it? Until around 125 days. What else can we see? Well, also there's not a huge difference between the farmland here and that, so there's similar mean numbers to farmland, right? But the other thing as well is we're looking at, again, just sort of evaluating all of this. If we're looking at the figure alone, we cannot conclude how well this is biodiversity-wise, because we don't know how many of each You know, we only know the mean bee species richness. We also only know the mean number of bees. We do not know how many of each species there are. So we don't have the index of diversity in this figure. Or we don't, or you could say like the number of each species has not been measured. Again, it's looking at this figure and thinking what the limitations are of it. There's nothing wrong with their method because, I mean, you can assume that scientists do a method right. So there's no errors of like, for example, I don't know, reading a beaker wrong or something. However, they've not told us enough information to conclude things like, oh, there might be something else going on here, like predation, blah, blah, blah. Like, they're asking you here to look at this figure and say, from this figure, can you say that? You need to look at it and say, well, this figure shows this, or yes, but this figure doesn't show this, and I need this to make this conclusion. Right? This is only telling me how many different species there are and how many bees they collected. It doesn't tell me how much of each one there is. So I cannot sit here and say, well, you know, yeah, I mean, the natural habitat is the best. You have to look at it at very face value. So scientists collected bees using a method that was ethical and allowed them to accurately identify which one they belonged to. So just one consideration they must have taken to make sure that it was ethical. Well, I mean, you know, not harm them, but also allowed the bees to just kind of return to normal life, you could also say. But then allow them to identify accurately the species to which they belong. So then you're saying, as in, how can you figure out a good way of working it out, like what they are? So it's something around the lines of being able to look at them up close. So for example, you could, I don't know, use like a what's it called, a booklet or like a reference to identify bees species. Or for example, you could, I don't know, look at like pictures or something like that. The question's asking you to identify the species that they belong to, not to identify what they recaptured. I think a lot of people kind of were like, oh, put UV light on it and then you can look at this, blah, blah, blah. It's like, no, it's saying, how can the scientists look at this and know what species is? So in another context, you might see this as in, for example, quadrats. If you're measuring plants, you might see this plant and it's like, okay, well, how do I know what species this is? You look at a little book that's like, oh, this is this plant, rhododendron, whatever, right? So that's what this question's asking you. So remember to read the question carefully. And don't answer it based on what you think it's saying, answer it based on what it's actually saying. So now it says, suggest and explain 2 ways in which the scientists could have improved the method for this data collection. So remember, just think about your independent and your dependent variables, and then it gets quite easy. So in the independent variable, what are they doing? The biodiversity, so they're changing the habitat. The dependent variable is the biodiversity of bees. So then look at this and say, does the method match what they've done? So this is showing us the different habitats. So they all right with the independent variable. Biodiversity of bees, I don't see biodiversity of bees here. All I see is the mean number of bees and also the mean species richness. So no, we've not really collected that. What they're doing is they're looking at both of these to say, oh, does that match that? So, well, I mean, first of all, one of the things they definitely could have done is the number of individuals in each species. Why? It's so that they can calculate the index of diversity. A perfect experiment is where you're measuring directly your independent and your dependent variables. Here, they're not directly looking at that, and the question's told you here, but the data doesn't directly show that, so that's one thing they can do immediately. Then think about the method that they've done and other ways to get better data collection. Well, they've done it what? They've collected it at so they did it 4 times per year. You could increase the number of times they collect bees so that they can have a better line of fit. Because, for example, if they did it like, I don't know, here, 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 and here, they're assuming that it's like this. What if in between there was a dip like that and then it goes up? You don't know that. So doing more intervals is always good. And then again, what other methods can you think of? Well, also as well as that, they could also do more sites as well to see if there's some more, like, you know, more natural sites or more town sites or saying what they actually are to get more accurate data. So that's, again, just always think independent, dependent, do they match? So now it says 3 of the bee species collected in the farmland areas with this, this, and this. What do these names suggest about the evolutionary relationships between these, and explain your answers? So evolutionary relationships is all to do with their, you know, phylogeny and all that stuff, right? So desperate King Philip cried out for goodness sake. So, you know, domain, kingdom, phylum, class, order, for goodness sake, family, genus, species, right? So whenever we're reporting the name of species, we use a binomial nomenclature, which basically means we're going to look at where you do their genus, followed by their species. So remember that this is a hierarchy. So you've got like the domain, the kingdom, and so forth, blah, 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 blah. Each one of them is non-overlapping within each other, right? So for these both have the same genus. So they must have come from the same similar common ancestor to go down that route. So if you think about it, it's like branching off like this, right? Whereas this one's a different genus. So already off the bat, you can say, well, the what is that? Chlora, blah, 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 and the other A, pipa, whatever, blah, 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 are more closely related because they exist in the same genus, right? So for example, if I look at chimpanzees, their genus is Pan troglodytes. I know, quite funny name, I know. Whereas, what is it? Humans are Homo sapiens. We're from different genuses, right? But if we go a bit further, if we go back up to family, the family is Hominidae, right? So we have the same family as chimpanzees. Different genus and different species, but same family. So that's where we're evolutionarily related to them. So this one says formation of an enzyme substrate complex increases the rate of the reaction. Explain how. So it's saying that when you get an ES complex, you somehow get an increased rate of reaction. So the only thing that explains that is it decreases the activation energy, right? But you need to say how. You can't just jump in and say, oh, you know, it's substrate and then it binds to the active site, blah, 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 blah. It's not asking that. They've already made an enzyme. They're literally asking you, enzyme substrate complex increases reaction rate. So they don't care about anything beyond that point because it's just asking about those two things. So the activation energy is decreased. You need to think how. You can't just say by providing an alternate reaction rate. That's like every catalyst in the universe. What they do is that they stress bonds, or you could say they weaken bonds, they bend bonds, they put tension on them. That's how they work. Catalysts, for example, things that join things together, hold them together so that any repulsion between them stops. Or if it wants to break them, it stresses the bonds between them and breaks them apart, like these enzymes. So that's what the question's asking. Please read the question. You don't need to yap about all that other stuff. A scientist measured the rate of amino acid removal from a polypeptide with and without an enzyme. With enzymes, 578 amino acids released per second. Without, 3 times 10 to the minus 9 amino acids released per second. Calculate by how many times the rate of reaction is greater with the enzyme present, and then give your answer in standard form. So with the enzyme present, it was 578 amino acids per second. Without an enzyme, it was 3 times 10 to the minus 9. So to work out how many times more it is, it's that divided by the other thing. So 578 divided by 3 times 10 to the minus 9 gives you 1.9267 times 10 to the 11, which is, and you've got to follow this number of decimal places or significant figures that they give you. So in this case, 3, so 1.93 times 10 to the 11. So then it says, another scientist investigated an enzyme that catalyzes this reaction, so it's ATP to ADP plus PI. They set up 2 experiments. So C used the enzymes and different concentrations of ATP. So they're dependent and independent, remember? So their independent is concentration of ATP, right? The enzyme's the same. Dependent is whatever they've ended up recording, which is what? The rate of reaction, so the rate. L has the enzyme, a different concentration of ATP, but also Lixose as well. So the independent is going to be the concentration of ATP still, and this is the dependent is still going to be the rate, right? But if you look between these two together, like if you factor in both of them, the dependent of the overall thing and the independent of the overall thing, Lixose, or Lixose or whatever, and the dependent is the rate between the two of them. So can you see how there's like sub-rates and stuff? So just kind of make sense of that and it gets a lot easier. So it says, the scientists measure the rate of reaction between each experiment. The results are shown in 5. So again, just have a quick look at what's happening line by line. So line L, the rate of reaction increases with time until about like 1 point something, 2 something, or 3 something, and then it kind of starts to level off and it becomes constant. Whereas in C, it kind of increases a little slower, less steeply, and levels off at a lower point than L, right? And then compare the two of them. So at the start, this one's increasing faster. The rate of reaction is going increasing quicker in a higher concentration of ATP, whereas here it's increasing more slowly, and this levels off at a higher point, and this levels off at a lower point. So just quickly mentally skim out what's happening there. Now the question says, calculate the rate of reaction of the enzyme activity with no Lixose of 2.5 moles per millimoles per decimeter cubed of ATP as a percentage of the maximum rate shown with Lixose. So the rate of 2.5 at C, sorry, the 2.5 moles at C is going to be what? So here to here, that's 0.41. So 0.4, 0.6 minus 0.4 divided by 5. 0.4 plus that is 0.44. The max rate of L is 1.4. So 0.44 divided by 1.4 gives you, oh sorry, and times it by 100 we should do because we're getting a percentage, gives you 31.4%. There you go. So it says Lixose, whatever it's called, binds to the enzyme. Suggest a reason for the difference in the reaction shown in figure 5 with and without Lixose. The amount, I can already predict this, that people have probably started yapping about enzyme substrate inhibitors and this and that, blah, 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 and completely fumbling the question. And I mean, I can see in the mark scheme, it tells you to ignore it straight away. And I can also see in the examiner report that lots of people started just yapping about it being a, you know, an inhibitor, this and that, blah, 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 changing the shape, this and that, yada, 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 yada. Which is, like, come on, man. Don't just answer the question based on what you think it's saying. Look at what it's telling you. If you have to scribble it out, then fine. That's what I sometimes have to do. So Lixose binds to enzyme And this leads to what? So it says suggest a reason for the difference in the results. Well, it shows you that there, on these results, that you have a higher maximum rate of reaction. They want you to explain why Lixos binding to an enzyme increases the rate of the reaction. Okay? That's what the question's asking. That's how you figure out. It's what the text of the question says. So you need to think in your head, what do I know about this? So obviously loads of people like to be like, oh bro, clearly it must be an inhibitor, blah, 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 blah. Like, okay, well, it's not inhibiting here, is it, if it's going higher? So we need to think of possible reasons why. So it tells you Lixos binds to this enzyme and for some reason the rate goes higher. If we have a higher rate, that must mean that there are more enzyme substrate complexes because, and then we need to think about why. So if there's more enzyme substrate complexes, then it must mean there's more successful collisions. It's not possible any other way for it to go faster. Then you need to think, well, okay, well, how could this be? Why is Lixos binding to this probably making that happen? Maybe it's the only possible explanation for that is that it's got to. Remember, these collisions depend on the active site colliding with the enzyme substrate. So if there's more enzyme substrate complexes and the substrate's staying the same, it must be that the enzyme's better at fitting at it. So it must be that the active site changes shape to better complement or be complementary to the substrate. There's literally no other possible explanation based on the text alone. Other people started yapping about respiratory substrate, blah, 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 blah. Like, it literally just says, how does this lead to this? Answer that. So it says, draw the structure of an amino acid. So every single amino acid has an amino acid, carboxylic acid. So by default, it's going to have COOH, which is the carboxylic acid part, and H2N, which is the amino part. Every single one of them has an H down here, and what separates them is the R group up there. There you go. So table 1 shows mRNA codons of the amino acids made for each codon. It also shows some of the R groups of the amino acids as well, the properties. So this is your first base, U, C, G, A, G. So if you have the first base starting as that, it can be any of these. Then if your second base is U, it could be either this or this. And then if your third base is U or C, then it's going to be this. Or if it's A or G, it's this. So that's how you interpret that. And then it tells you the properties of the amino acid is that all these grey ones, no overall charge. Darker, slightly darker is positive, and then dark is negative. Fine. So the question says the genetic code is degenerate. What is meant by that? So degenerate means, anyone? It means that more than one codon or base triplet makes the same codes for the same amino acid. So then all you do is you just pick an example. So I don't know, we could go with U and then UUC. UUU and UUC make phi. There you go. So scientists investigate the changes in amino acid sequence on of a human enzyme resulting in mutations. All of these amino acid changes result from single base substitution reactions, and the enzyme is a polypeptide that is 465 amino acids long. Real quick, independent, dependent. So they're looking at the changes in the amino acid sequence resulting from mutations. So the independent, technically speaking, is going to be the mutations, because that's the base sequence, and the dependent is the amino acids that come from it. Just helpful to get a good idea of that. Table 2 shows the results of 3 base substitutions. We got amino acid number 203 in this thing. That's 465 long. 465 long, and this is what's happened, blah, blah, blah. So then it says, what is the minimum number of bases in the gene coding for this polypeptide? So it's 465 amino acids long. One triplet makes one amino acid. So if we've got 465 amino acids, 465 amino acids, then we're going to have 3 times that. 465 times 3, that gives you 1395. It says in the examiner report, a lot of people just overanalyze this and added like start and stop codons and this and that and doubled it because it double-stranded, blah, blah, blah. Like it just says there, the minimum number, you bare minimum need this. Yes, the other things are technically correct, but like the absolute core minimum, there's got to be this much. Like just go with the question. Use information for table 1 to tick one box that shows a base substitution that would result in Val-Dala at this amino acid number 203. So then we look here, we've got Val-Dala here, but the problem is that if we swapped like U and C, then it's different, because it doesn't matter what these and these you have, it's U and C that switch. So none of these in the middle have a U to a C switch that fits this, because you can see here that does have a GUU, and that is a GCA, right? So you might be misled into thinking that, but the question's saying the mutation in D. So it's going to be the opposites, all of these. So if we have, like for example, I don't know, GUU to whatever, then we're going to have to, it's going to be the opposites. So it's going to be C. So if we were looking at this GUU one, it's going to be CAA becomes CGA instead. CGA, sorry. And also, if it's DNA, this can't be it. This can't be it already. So there you go. A change of glueless amino acid 300 had no effect on the rate of reaction catalyzed by the enzyme. The same change at 279 significantly reduced the rate of reaction catalyzed by the enzyme. Use all this information in your knowledge of protein structure to suggest reasons why the difference of these two changes. So every single, like, huge amounts of people fumbled this. Everyone was talking about things like amino acid sequence and frame shift mutations and this and that, blah, blah, blah. And then they were, like, talking about all kinds of things, saying just shape of active site, 3D structure of protein, rather than saying tertiary structure. Just a lot of problems here, and just fumbling. Again, it can all be resolved by simply just reading the question and answering what the question is telling you, not just trying to, you know, blurt out words that you think it's saying. The question has told you that change at 300 amino acid, you know, like from GLU to LYS, no effect on enzyme. But then it's told you, but if the same thing happens at 279, significant, you know, enzyme activity. So you need to think about why. It must be, but if there's a change here and there's no drop in the enzyme function, it must mean that the change at 300 did not affect the active site. You can't just say 3D structure. That means nothing. It is the active site. Otherwise, the enzyme wouldn't work, right? However, the change at 279 may have changed the active site. There's literally no other reason why the enzyme activity would drop. And then you need to say how, by, and then we need to think, well, okay, the active site depends on what? It depends on the tertiary structure. Tertiary structure is all to do with ionic bonds, hydrogen bonds, and disulfide bridges. So it must have changed, because it's changed the tertiary structures, hydrogen, ionic, or disulfide bonds, right? Otherwise, there's literally no other explanation for this. And then why is that this change doing that? We need to look at the table. So if you look at GLU, wherever it is, so that GLU is negatively charged, whereas LYS is positive. So that's probably by how it's changed. It's probably changed your ionic bonds. So I didn't have to talk about, you know, silent mutations, frameshift mutations, this or that, blah, blah, blah. You know, like, it all is in the question. Don't answer the question because you think, oh, I think it's on this topic, I'm going to say this. Literally, like, step by step it. Figure 6 shows a faulty form of meiosis that can happen in some plants. So meiosis, remember, is you're making 4 genetically different haploid cells. Haploid. So here, it's told you in the question it is diploid. So then this and this are homologous pairs. So that must be like, I'm going to say maternal and paternal. I know they're not really got mum or dad per se. And that's going to be maternal and paternal. So those are a homologous pair. You know, when I was yapping about it in part 1. Question 1, sorry. The first division of meiosis, can you see how we have 2 sister chromatids joined together by a centromere? These are not a homologous pair. This is the same, like, maternal-maternal and maternal-maternal copied here. And this is a separate chromosome. Can you see? So it could be like, I don't know, maternal-maternal, paternal-paternal, but they're not mixed. Can you see how the homologous chromosomes have separated? The maternal and maternal have separated, and the paternal and paternal have separated. In this division, then, what must happen is those chromatids have separated, because you can see now they're single-stranded. So if we look at what's happened here, we've got 2 big long ones, and then 2 little shorter ones. And they've told you in the question that there's 2 daughter cells that do not develop, because these all have to be divvied up. But can you see how there's 2 big long ones here, 2 short ones here, 2 big long ones here, 2 short ones here? Because there's 2 copies of each chromosome, it is diploid again, which is wrong. So then it says complete figure 7 to show what would normally happen, basically. Draw what normally happens. It says here's the chromosomes after the first division of meiosis. So in your first division of meiosis, it's going to be short, like this, and then this. And then the chromatids separate in the second one. So each one of these should have this. 1 short, 1 long, 1 short, 1 long, 1 short, 1 long, 1 short, 1 long. The homologous pairs have already been separated. That and that are homologous pairs. These chromatids then separate. So if 2 diploid gametes fuse at fertilization, it can cause the growth of a tetraploid plant which has 4 copies. So it's actually got 4n as its number. Red clover is a plant grown to produce cattle feed. Tetraploid clover plants produce a higher yield than diploid. Whether a red clover plant produces 2n gametes is genetically controlled. Okay, this will be interesting. So what they did is they investigated the possibility of breeding red clover plants that only produce 2n gametes. In the cycle 0, they identified 2 end gametes. In the breeding cycle 1, they used the gametes producing them to make offspring. So what they did is they did 2n and 2n. In this case, they just grew red co-equivalents and picked out the ones that have 2 ends. In breeding cycles 2 and 3, they then looked at which ones had 2n gametes and then did 2n x 2n again. And then here's the table. So in breeding cycle 0, 1, 2, and 3, we can see 50 didn't and so forth, whatever, blah, blah, blah. And it says, here's the null hypothesis. The proportion of plants that produce 2n gametes will not change from one to the next. Use table 3 to show the expected numbers that did not produce 2n gametes and expected numbers that did produce 2n gametes after one cycle. So they're saying that the proportion, so it doesn't have to be a specific number, it's the proportion of 2n gamete making things doesn't change. So in this case, in total, we have 54 in total. 50 of them did not, and then 4 of them did. So that means 50 out of 54 is the proportion. So 50 out of 54 is whatever, right? And then the rest is this. So here, in total, there are what? So 14 plus 42 is 56 in total now. So if you would do this times 56 to find the proportion in this one. So 50 over 54 times 56 gives you 51.8, so 52. And then the proportion here is 4 out of 54. And we have 56 in total, so then you would do that times 56, which gives you 4. So the proportion is the same, because this is each generation. So these are the proportions. So then they said the scientists tested the null hypothesis using the chi-squared statistical test, and then after one cycle, they calculated the chi-square value of this. The critical value at p is this. What does this suggest about the difference between the observed and expected results, and what can they conclude? So one thing that people always yap about and completely screw up are these questions to do with statistical tests, and everyone just says like, oh, the results are not due to chance. You can't say the results, you can't just throw words in and try and make model answers. It literally never works. Rule number 1 is, what is the chi-squared and whatever, the p-value, actually testing? What is it actually looking at? The p-value here, it's told you, is the testing the null hypothesis, blah, 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 after one like this. The critical value is 3.841. This chi-squared test is testing the differences between the observed and expected. So it's testing the differences seen in observed versus expected. Not the results. You don't get any marks if you don't say that. Now this p-value, so then we need to think about what the p-value actually is. They've told you the critical value at this is this. When your chi-squared value that you calculate is higher than the critical value, then it's this or less, the probability. So the critical value is 350, which is greater than 3.841. Therefore, the probability of, and then what it's testing, the differences in observed versus expected results is not due to chance. Therefore, because then it says, what can they what can they therefore conclude? Therefore, their null hypothesis is that there is no difference. We just looked at them and said there is a difference, and it's not due to chance. Therefore, they can reject the null hypothesis. Questions don't... I've seen people just write about null hypotheses when there isn't even one, and then they just talk about all kinds of stuff. The p-value, look at what it's testing, and then look at their hypothesis. They're saying there's no difference. Well, we just said this p-value says there is a difference that's not due to chance. Therefore, they can reject it. So again, rules with this is everything. No matter it's a t-test, look at differences between means. Correlation coefficients look for the correlation between two things. And chi-squared is a between observed and expected results. The p-value in this question is testing the differences in the observed versus the expecteds. So therefore, anything you're saying about p-value being significant or the results is this. So here now it says, use your knowledge of directional selection to explain the results in table 3. Directional selection is where, imagine you've got your normal bell curve chilling, right? Directional means you're favoring one extreme. So this could be, I don't know, like the number of individuals. This could be, I don't know, like height. Let's say we have this weird environment where for some reason you're short, and if you're tall, you're favored. So directional means all the population gets skewed, the mean and the middles and stuff, all get skewed towards the higher end. In another population, it might be that it's actually more advantageous to be smaller, so you get skewed that way. Directional means it's towards an extreme. So stabilizing is different, in which you are favoring something that is stable. You're not going to an extreme, so you might be that the average height is the best. And disruptive is where either of the extremes are quite beneficial. So the middle is what gets screwed over the most. In this question, they're talking about directional selection. So we need to see, is there a direction that things are heading towards? Whenever you're looking at selection and evolution as well, this is all about changes in allele frequency. The whole thing to do with evolution is changing allele frequencies. So in this circumstance, the number, they've told you in the question that the things that, so whether it produces 2n gametes or not is genetically controlled, as in there is an allele to this. Here, you can see that the numbers that did not produce 2n gametes was 50, 14, 2, and 0. That's what they would expect. Sorry, observed. The expected of these. You can see that the number that did not produce them went down, whereas the number that did went up. So the direction it's going towards is that more of them are having 2n's. So a lot of people like to say every single step and whatever. Try and say it the other way around. So there's more, each generation has increasing proportions of 2n gametes. Right, but that doesn't explain changing allele frequencies because it's a selection. Therefore, it must mean that there is more, the allele frequency, the amount it's appearing in a population, the allele frequency for 2n gametes is increasing, right? And we need to think about why. Why is there selection happening? So a lot of people talk about survival, reproduction, this and that, blah, blah, blah. They're not surviving or reproducing. The scientists literally made them breed and do it then like that. So there's no selection pressures here except other than just the breeding, sorry. So then the reason it's happening is because for some reason, the ones that are making the 2n gametes are better and more likely to reproduce and this and that, blah, blah, blah. The question has literally told you that the scientists have picked 2n producing plants. The selection pressure is the scientists saying, okay, you guys get to breed, you guys don't. So the reason this is happening is because scientists chose and bred 2n gamete making plants. And therefore, then what they did is they reproduce, because there's no survival stuff here really, and then they pass on the 2n allele gametes. And then its population increases. So again, directional selection, as you're moving towards one thing, it's all to do with changing allele frequencies. So you've got to think about what's making them happen. In this case, they are not surviving and reproducing and blah, 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 blah. The surviving and reproducing is not happening here. It's the fact that these lot are just getting a chance to, you know, procreate. There's nothing to do with the selective advantage here. So you got to look at the question and stop saying model answers. I hate model answers, bro. So question says, when a person is bitten by a venomous snake, it injects a toxin into the person. Anti-venom is then injected as a treatment. So anti-venom contains antibodies against the toxin. So here's our toxin. Here's your anti-venom. And in this case, actually, it's an antibody. So there's my anti-venom antibody, because they've told you in the question. This treatment is an example of passive immunity. Explain how the treatment with anti-venom works and why it's essential to use passive immunity rather than active. So let's talk about how it first works. So only 28% of people manage to actually get this. Literally writing down and working what the question says. The anti-venom is antibody. So the antibody binds to the toxin. The toxin is the antigen, and then eventually the toxin is destroyed. And then, why passive over immunity? Sorry, but why are passive over active? Well, let's have a think, shall we? So you compare them in your head. Active versus passive. So a lot of people are talking about the fact that for some reason, you know, you can make memory cells, this, blah, 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 blah. It's not that. It's why would you choose this over that? Well, active immunity is slower. You'll have to wait for phagocytosis to happen, for T cells to be stimulated, for B cells to do clonal selection, plasma cells, blah, 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 blah. Like, we need to treat them now. So we give them passive immunity because the antibodies are right there, going to town, going there. You cannot just say passive immunity is fast. It has to be faster. It's a comparative thing. So here, figure 8 shows the procedure to produce anti-venom. So what they did is they get one species, they collect their venom, they get a bunch of venom mixtures. Then what they do is they select a bunch of animals, they quarantine them with a vet, then they inject the venom into an animal, they collect the animal's blood, they have this vet checking it's all okay, and then they take antibodies from the animal blood plasma. Fine. A mixture of venoms from several snakes in the same species is used. Suggest why. So, same species, right? You would expect there would be the same venom. But if they're using a mixture from several snakes of the same species, there must be some difference between the venom that they're making. So first of all, otherwise what's the point? Why can't you just use one snake? So there must be different forms of the same venom or toxin. So if that's the case, the reason that must be, and that makes sense because, you know, different alleles, hello? Like I have black hair, some people have blonde hair. We still have hair. In this case, they still have venom. It's just a different side, you know, form. And that can be as a result of mutations in alleles. But that still doesn't say why they're taking a mixture of it and actually using it. It's because if you have, well, different versions of it, then you're going to need different antibodies in the response. So there's going to be different antibodies complementary to the venoms or venom toxins or whatever. Like they're all the same toxin, just slightly different. So we're going to have different antibodies. That's what it's trying to get at. A lot of people are saying it needs to be to identify the right species. Like, no, they've told you that they're using a mixture of different venoms of the same species to make this stuff. So there's only one explanation, which is that. So horses or rabbits can be used to produce antivenoms. When taking animals' bloods to extract them, 13 centimeters cubed is collected per kilogram of the mass. So 13 centimeters cubed per kilogram is taken. The mean mass of horses is 350. The mean mass of rabbits is 2. Using this information, which would be better for the production of antivenoms, and use a calculation. So for horse, if we get 13 for every kilogram, then we've got 350. So it's 13 times 350, which is 4550 centimeters cubed of blood needed. Whereas a rabbit is going to be 13 times 2, which is 26 centimeters cubed blood needed or collected. So using this, when taking the extract antibody, blah, 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 blah. So in this case, you can get a lot more blood out of the horses. So you would say horses, because you can get more blood, so there must be more antibodies. So then therefore, a reason being is because there's this and this. So that's what you can write. I'm just going to skip ahead and do the next bit. So then it says, during the production, they had ongoing observation by a vet. Suggest one reason why. So look at it and think about what's going on. You get animals, you observe them with a vet, they get vaccinated, and you still have them observed via a vet. So the only reason that I could think at this point, and in the examiner report says too many generalized answers were here, I mean, you're injecting them with a toxin. So it's to ensure the animal, like, I don't know, doesn't die? The animal doesn't, I don't know, suffer from the toxin because it doesn't die, because it doesn't lose a lot of blood because you're taking a lot of blood from them. Or that, I don't know, it doesn't have a pathogen that could be stuck in humans as well. So, for example, if this animal's infected with something else that can go into humans, and you take blood from them, and now you're in antibodies to that, you're now putting an extra disease into humans. As well as that, you could also talk about accepting ethical or legal requirements as well, but I mean, that's a bit of a point. So in this question, only 7.3% of people that did this could get like an actually good answer. It says during vaccination, each animal is injected with a small volume of venom. Then 2 weeks later, they use the same, but they do a larger volume of venom. It says use your knowledge of the humoral immune response to explain this vaccination program. Just as a real summary, you get in the immune response, once the phagocyte engulfs and digests, blah, 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 phagocytosis, it will interact with T cells, so helpers T cells, and what they will do is they will stimulate cytotoxic T cells, more phagocytes, and then also more helper T cells. The interactions of all of these cells with each other is your cellular response. And also, sorry, as well, it stimulates B cells too. So those B cells that are stimulated will undergo clonal selection. So only one of them that's complementary to the antibody, the antibody they make that's complementary to the antigen here, and that was picked up to start with, is selected. So you get clonal selection, divides by mitosis, you get plasma cells, and then you get monoclonal antibodies. So this whole thing is the humoral response. Humor kind of means like flowy, juicy stuff. So cellular is the interactions between each cell. Humoral is like producing lots of humor. Monoclonal antibodies. So this question is saying to discuss the humoral response to explain why they've chosen to inject a small amount to start with, then a bigger one. So you look in here and you're thinking, okay, well, what's going on with B cells? What's going on with this? What's going on with this? And all of this. You should be thinking, oh wait, if I inject a small amount to start with, then another one another time, then they're going to make more antibodies in a bigger and larger volume. And that's exactly what happens. So the first injection, you have your cellular response, blah, blah, blah. And then what happens is that B cells that are specific to the venom undergo clonal selection. Because remember, you don't want every single B cell. You only want the ones that will make antibodies for that, or else you just waste all your energy. Undergo clonal selection, they divide by mitosis, and they form plasma cells. Some of those will go on to become memory B cells as well. So then, once you get to the second injection, these memory B cells are activated. And what you get is you suddenly get a very large, fast, and large increase of monoclonal antibodies produced. And that's exactly it. That's where, remember, we're looking to explain why they're using this vaccination program in making a supply of antibodies that we can inject in people who've been bitten by this snake. So ideally, you want to max out as much as you can, and that's why they do this. So scientists investigate the effect of a heat treatment on mass transport in barley plants. So again, like the dependent variable they're looking at is mass transport. Their independent is heat treatment versus none. I know it seems a bit over the top, but it really helps you sort out your thoughts when you're looking through this. Now again, we can't look at mass transport directly, so the dependent variable and actual independent variables in this are probably going to be different, right? So that's what this part's telling us, but it's probably different throughout. So what they did is they put steam on one side of the leaf, so one short section. This area is shown in the arrows in figure 9. So the arrows in figure 9. So that's been heat treated. Let me do it in a different color. That's been heat treated. That's been heat treated. Fine. And then what did they do? They did not apply steam to the leaves of the control plants. That makes sense, as we're checking steam versus no steam. They then gave CO2 containing radioactively labeled carbon to the plant in the area shown in the rectangle boxes. After 4 hours, they found the position of the radio, blah, 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 blah. These results are shown in figure 9. They recorded the water content of the plants of the leaves that supplied radioactive, blah, 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 and the results are shown in table 4. So then in this circumstance, the dependent variable here, mass transport, is actually the movement of the radioactive carbon, which in turn is what they're using as a marker of mass transport. So if we look at the figure here, we can see what? So 0 hours, 4 hours, it's darkened, but this has gone a lot brighter, so it suggests it's all kind of gone there. Whereas here it's, you know, the rest of it still has a bit going on. Versus here, you know, there's like some and nothing. And here, there's like some all over and still some all over, but there's a bit more here and here. Fine. Don't know anything about it yet. Then it says, here's the water content of the leaf from which the leaf was taken. So the heat-treated plant had this much water in it. The control plant had this much. And if we look, they're plus or minus 2 standard deviations. There is a bit of overlap between the two of them, because that can go all the way up to like 95-ish. Then it says the scientists concluded that heat treatment damaged the phloem. And it says, explain how figure 9 supports this conclusion. So in this question, remember that they're using the movement of this radioactive carbon to look at mass transport, which happens in the phloem, which is why I say you should do that. So if you look at figure 9, what's going on here then? So in the control, without any heat stuff, you've got some, you know, after 4 hours, this starts to glow more. So there's more radioactive carbon here. But as well as that, there's a bit of glow in here versus here. So there's some more radioactive carbon here as well. Whereas in this one, there's all glow in here, but there's none of it here. So the radioactive carbon, well, I mean, it was here before, now it's not there, it's all gone. So what must have happened is that whatever the radioactive carbon was in has been used up here, and none of it could get out from here down there. It's been choked there. Whereas here, it's still spreading nice and evenly, so it's pretty happy. So in A, you know, the radioactive C is now absent in the bottom part. So you need to think, where could carbon go inside a plant? Why has it disappeared? Well, it must have been, you know, used up into something. So it must have been, I don't know, converted. Because what did they do? They provided carbon dioxide. So it must have been converted into glucose during photosynthesis, right? But that doesn't explain why it's disappeared, because surely it's still there. It should still be there. But if we look, we can see that it's now really concentrated up here. So all of it that's made in photosynthesis, remember those are like the roots, must have all accumulated here and actually can't get out. So in B, if we look, it's nice and evenly spread out. So it must mean that this, you know, this radioactive C was, you know, was able to translocate throughout the plant. Whereas in A, it couldn't. And you need to think, well, why is that a problem? Well, it must be because the heat treatment damaged, like, for example, companion cells, all that kind of stuff. So living cells, because remember, phloem requires companion cells to do active transport. Cells, and you know, what is it? The phloem movement needs ATP. So what might have happened is that this heat treatment might have stopped respiration, because that requires enzymes, and it wasn't able to do the ATP, and it's all stuck there. So the scientist also concluded that the heat treatment did not affect the xylem. Explain how the results support this. So don't just go yapping about everything else, because I can see already that here there's just people saying all kinds of stuff. Look at the table. A table is, they've taken leaves from here versus here, and they've looked at the water content. So the water content is what their measure of the xylem stuff is. Now, I said before, there's no overlap in the SDs. You cannot just say, oh, their results don't overlap. Like I said before with the p-value in the chi-squared question before, what is the SD testing? Or what is the SD related to? Like, what does it actually mean? It's looking at, well, it's the water potential, the water content of a leaf, right? However, let's cut out the search just. So it's looking at the water content of the leaf, right? To start by describing what's going on, well, the SDs overlap in, you know, whatever A and B. Therefore, the water content difference must have been due to chance. So if that's the case, then there's therefore no difference. So then you need to explain what that means. If there's no difference in water content, then the xylem must be unaffected because the water's still able to get around where it needs to be. Because they're looking at the leaves, because remember in leaves, transpiration happens, H2O comes out. In your xylem, you have this column of water that gets dragged up as water's coming out from the leaves. So that's why they've done that. Here, a lot of people did not realize that, like, the water moves into the leaf from the xylem, which is why we can say that the xylem must have been unaffected. The scientists then investigate the movement of iron ions from the soil to young and old leaves of heat-treated plant barley plants into the leaves that were not heat-treated. The scientists then investigated the movement of Fe3+ ions from the soil to old and young leaves of heat-treated barley plants and to the leaves of plants that were not heat-treated. Heat treatment was applied halfway up the trees. The scientists determined the concentration of Fe3+ in the top and lower halves of the leaves of each plant. Their results are shown in this table. Figure. So in this figure, again, always look at things one by one. So on the y-axis, I can see it's a ratio of Fe3+ in the top half. So Fe3+ in the top to the lower, right? And then here, I've got old leaves and young leaves. So then as well as that, the dark gray is untreated, the brightest treatment has been given heat treatment. So compare one by one. So I'm going to look at the half treatment halfway up the leaf. So in the old leaves, heat treatment had this, like a 0.4, you know, between 0.4 and 0.5. So there was less Fe on the top than in the lower, because if, remember, this is a ratio, you could rewrite a ratio as a fraction, and that might help visualize it, so it's top over lower. So if it's less than 1, it means that this is less than the bottom. But then in the young leaf, you can see that heat treatment gave you this. Fine. Doesn't mean that heat treatment directly causes them, because we're looking at old leaves and young leaves. So I'm doing it one by one. Then in the untreated, in the old leaves, untreated was higher on the top and the lower, but still top was less than lower, and then young leaf it was lower. So in the old leaf, if I heat treat them, there's slightly more Fe+ on the top than if I left them untreated. Whereas in the young leaves, if I heat treat them, there's less at the top, but if I leave them untreated, there's more, so there's opposites going on here. So then, it says here, what can you conclude about the movement of Fe3+ in barley plants? Use all the information provided. So clearly, number one, I mean, if we're going to look at all the information here that they've given us, is that again, heat treatment seems to have not really done very much difference-wise. It's actually higher with the heat treatment in the top half than the lower, whereas in the young, it's absolutely shot down. So one thing I can definitely say straight away is that heat treatment affects younger leaves more than older ones, right? So then the reason must be is that therefore heat must damage the phloem as a conclusion. However, if we're heat treating it, there's still, you know, things getting to the top. Because remember that they've heat treated upward half the leaf. There should be nothing if we're heat treating it. So as well as that, they've told you in the question that this is Fe in the soil and this is the leaves. So it's moving up through the plant. So that's also how I know it's the phloem. However, we can still see that these two still have some in the top half of the leaf than the lower. So there definitely must be still some movement of the Fe3+, but we've damaged the phloem. So therefore, we can also say that the xylem must, that's the only other way of moving things up a plant, also transport Fe3+, and it must be more in older plants than younger, because you can see as well that, like, despite heat treatment, there's still a decent amount of it going on. And for there to be such a significant difference, even though there's heat treatment, it must be that there's something else, you know, the other thing that's used for transport is also not working. As well as that, all of these ratios are less than 1, so I can go ahead and say, well, also all Fe3+ is less in the top leaf than lower, because all ratios are less than 1. And also I can say, well, there must be, you know, more Fe3+ at the top in older than younger. What else can you conclude using all the information? So if we look at everything here, I can't just go ahead and say all of this because I don't know anything about how much there was. And also I know nothing about statistical tests. It's asking you, what can you conclude from this? I've not got the guts to say that that's 100% true because there's no statistical test. But you need to say why to see if differences observed are significant. When it says to use all the information, it means literally like what you see here is what you're concluding. If there's no stats test, you should mention that because you shouldn't really be interpreting results of these tests without a stats test. Otherwise you don't know if it's a valid and it's a significant test, blah, blah, blah. So it says describe the role of two named enzymes in the process of semi-conservative replication. The examiner report says only about 25% actually got like the right amount of marks because a lot of people were just yapping about all kinds of garbage. So here, one of the enzymes is DNA helicase, and what it does is it breaks hydrogen bonds between complementary base pairs. Many people were saying that it hydrolyzes them. Hydrolyzing is putting a molecule of water in. These hydrogen bonds are not having a molecule of water put in, so no. The other enzyme that's doing the job is DNA polymerase. And the other thing this is something I see a load of people fumble as well, and it really, really annoys me, is that what it does is it joins adjacent nucleotides via condensation reactions to form, and I'm just going to write PDE because I'm lazy, but you write the whole thing, phosphodiester bonds. People were saying that it joins complementary bases, which is incorrect. If I have a strand which is ATCG, right? That's one strand. The other strand is going to be complementary, so it's going to be TAGC. These are the hydrogen bonds between them. They're not that, like, strong. These are your phosphodiester bonds between each of these. DNA polymerase makes the polymer of DNA. The polymer of DNA is one of those. That's one strand. It's joining the nucleotides next to each other. Because what people are saying is that it's joining these two together, you would end up with just sticks of A to stuck to a T, and then a T stuck to an A. You wouldn't have a strand. You just literally have like awkwardly glued on things here. Or if we use the original as a template, you would literally just have A stuck to T, this and this and this. These won't even be joined, which is objectively wrong. And you can't just don't try and yap model answers that you got wrong, please. You don't even have to say like fancy words in this, man. Now it says, the scientists investigated the function of a eukaryotic cell protein called cyclic A. It's thought to be involved in the binding of one of the enzymes required at the start of DNA replication. So what they did is they treated cultural cells in the following ways. So what they did is they got controlled cells, they added an antibody that binds to cyclin A, they added RNA that prevents the translation of it, and then they added RNA that prevents the translation of it, and then also added cyclin A protein. So effectively what they're doing is their independent is cyclin A versus none, and their dependent is what are they measuring? So then determined, well, there you go, percentage of cells with DNA replicating. And here's their results. So in the control, it's 91%. If they're adding this antibody that binds to cyclin A, we can see that the percentage of cells is decreasing. In E, they added the RNA that stops it from happening. We can see that also decreases. So these two are suggesting that it's being bashed. However, if you add RNA that prevents the cyclin A, we would expect it to drop down. But then they added cyclin A back in, so it's come back up. So that suggests that cyclin A is clearly somewhat important here. Because if we stop it and then add it back in again, it comes back to normal nearly. So then it says, suggest explanations for the results in table 5. So off the bat, we can say, well, that antibody, I'm going to write AB for short because I'm just keeping track of time, binds to cyclin A, so it cannot, contribute or initiate or whatever, it start DNA replication. That explains why it's down. Now, the other one is RNA. Well, it must clearly, it prevents the translation of it. So it clearly must be that it's stopping, translation. So it's stopping. It must be interfering with ribosomes making cyclin A protein. So it must be interfering with translation because mRNA comes out the nucleus, goes to ribosomes, blah, blah, blah. So it's the only explanation, really. So that's that. So that's explaining why they go down. You can, if you haven't noticed, sorry, as well, you should always try and do it in an order. So like I'm doing D, then I'm doing E, now we're on to F, sorry. So then in F, adding cyclin A back allows, DNA replication. But we're not done, because even then, if we look at D and E, why are they not 0? They should be 0 if it's entirely responsible to that. So we need to think about what might be going on here. Well, clearly, they're not 0. Some cells still continue DNA replication. We need to think about why. Well, if we look at what they've done in D and E, they added an antibody and RNA, so maybe they just had a little bit left over from before. So they could have had previously made or translated or whatever cyclin A that was left over that might have done it. Because it's the only explanation I can think of. And the question they've told you that like So this question then says, describe the gross structure of the human gas exchange system and how we breathe in and out. The mark schemes... Oh, redo the bit about the mark scheme that I was just saying, examiner report. The examiner report says that lots of people just straight up didn't talk about that. And very few people talked about the gross structure. And there were loads of answers talking about alveolar epithelium, gas exchange, tracheoles as well. Tracheoles aren't even in humans. And all kinds of absolute just yap. The question has asked, describe the gross structure of the human gas exchange system, and then how do we breathe in and out? So answer just that. So if we're looking at the gross structure, air enters the trachea, right? And the trachea divides into bronchi, and then they divide into bronchioles and so forth. Here's your big lung. And then it divides into alveoli. So you'd write that. It divides into left and right bronchi, which divide into bronchioles, which then divide into alveoli, where gas exchange happens. The question is not asking me to talk about gas exchange. It's literally just saying, describe the structure. Well, I've just done that. Next, it's how do we breathe? When we breathe, we want to make the volume of our thorax, the chest, bigger. It's a really hyperbolic example, but if we make it bigger, then the pressure decreases in the thorax. It's going to be lower than outside, so it drags air into it like this. Then when we breathe out, we want to squeeze all of that air so the pressure's high in here and shove it back out like that. So the way we do that is it's to do with the diaphragm and your external intercostal muscles. The diaphragm is the big one. So this process is called inspiration, which is breathing in. What happens is the diaphragm and external intercostals contract. What that then does is it increases the thorax, or the ribcage volume, and it will decrease the pressure. So then air enters the lungs via a pressure gradient. Then, when you breathe out, that is a passive process. It is the entire opposite of all of that. We don't forcibly breathe out. Imagine having to take a deep forced breath in every time and then another forced out. You'd be exhausted. So when you're breathing out, the diaphragm and external intercostals relax. It is the entire opposite. Then what happens is this increases the thorax's volume. And then it will, sorry, not increase, it decreases the thorax's volume, sorry. And it increases the pressure. So then air moves out of the lungs down a pressure gradient. If we were to do forced expiration, so if you take a really deep breath out now, you'll feel it in your chest. You'll be like, oh, that feels a bit gross. That's because of those internal intercostal muscles contracting. So when you're forced breathing out, then what happens is the same things, the diaphragm plus the external intercostals relax, but your internal intercostals contract. And then the same thing as what we said here, this and that, sorry, no, this and that, blah, 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 blah. You're forcing as much as you possibly can out. A lot of people also said stuff to do with saying, oh, the diaphragm flattens, but didn't say how. You can't just say things, you gotta like explain by every step. So there you go, nothing to the gas exchange here. So here it says, mucus produced by epithelial cells in the human gas exchange system contains triglycerides and phospholipids. Compare and contrast the structures of triglycerides versus phospholipids. I'm gonna just do TV for this and then PL. So what that means is compare them and say what's similar and what's different. So the similarities is that both have glycerol. They both have fatty acid. Like, right? Then that's right, that can be saturated or unsaturated. What other things do we know about them? Well, they're both made of fatty acids, both got glycerol, right? Another property of them that's key is that they both have ester bonds. You need to say where the fatty acids and the glycerol. And then what else? They're both insoluble. You could rinse those options easily. You could also say they both have C, H, and O, carbon, hydrogen, oxygen. Difference is, well, first of all, phospholipids have phosphate or phosphorus. Triglycerides do not. What else do they have? Well, they have one fatty acid replaced with a phosphate head. What else can I think of? So, oh, well, also that, and also, sorry, they have, so phospholipids have a hydrophilic head and hydrophobic tails, whereas triglycerides, remember, can you see how I'm saying comparative statements? This and that, this and that. Oh, this has been replaced. So triglycerides only have hydrophobic tails. And there you go. Like, it's a comparative statement. This and this, this and that, this and this. Every single thing that says compare and contrast, every single point has to have a comparative statement as well that is clear. So you can see I've said this has this does not, this does not. You need to do that because it even says in the examiner report, examiners do not infer links between separate statements. You have to make it clear. Other problems that were with this question is a lot of people didn't say what glycerol was. Other people were talking about energetic values. I'm not even too sure what they mean in this context. Probably something to do with it storing it. And all kinds of yap. All it's saying is do that, so do it. Next it says, mucus also contains glycoproteins. One of these is a polypeptide with the sugar lactose attached. Describe how lactose is formed, and then where in the cell it would be attached to a polypeptide to form a glycoprotein. So let's do number 1, which is how it's formed, and then number 2. Number 1 is how it's formed, so it's made of glucose plus galactose, joined by condensation, forming a glycosidic bond. In biology, always say what's happening, whether they're joining by condensation, hydrolysis, what bond is being made and what's being broken. Literally saying all of that is 3 marks. So if you just do it by default, you'll never miss any marks. I got nothing else to say there. So then it says, explain how to be attached to a polypeptide. The only thing that attaches things to polypeptides, proteins, is the Golgi apparatus. Or apparatus, whatever you call it. It's the Golgi apparatus attach lactose to a protein. Is literally it. Like, what else can you say? So then it says in the examiner report, lots of people talked about condensation reaction, but no one's, like, barely fused, you know. Like, a lot of people just straight up didn't say glycosidic bond. And then as well as that, a lot of people didn't realize that this is what's happening in the Golgi apparatus. So it's just like, you know, again, the question is just it. So there you go. Anyway, hope that helps. Thank you very much. Please like, share, and subscribe. You know the drill. And peace out.